Welcome back to the course Fundamentals of General Immunology. So today we'll try to discuss few interesting facts about the topics which we have covered till date, and those are all related to some of the things which are quite interesting in the field of immunology. So let's try to move on and see some of the facts which are very common in immunology cells as well as immunological concept. So when we discussed initially about the primary lymphoid organ, we discussed that we have thymus and bone marrow as a primary lymphoid organ in where we can see that there could be a production of T lymphocyte and B lymphocyte. While when we see origin of B cell, that comes from this Bursa of Fabricius. Now, Bursa of Fabricius is a very specific organ that's located in the dorsal side at the end of the alimentary canal and the dorsal side. So it is actually a very unique lymphoid organ in case of chickens. So these are the site of B cell production, the lymphoid organ, and in one side it is connected to the ileum, the intestinal part, and the other side it is connected to the outside opening which is cloaca. So essentially these are all very thick lining having a lumen, and there are a lot of folds which are present. These are actually if we take a cross section and look into that, those are all very rigid columnar epithelium. We also have a lamina propria. There could be submucosa, muscularis mucosa, and then serosa lining as well, which is typically any organ system. But then we do see that these are primary lymphoid organ in case of chickens and birds, and these are producing a B lymphocyte, and any deviation, any disformation or any kind of a problem in these particular organ can lead to immunosuppression. There is a disease called infectious bursal disease in case of poultry, and these are especially typical disease that is happening because of inflamed bursa. So the virus which is the causative agent for this disease is called infectious bursal disease virus. It's called IBDV. It's actually a virus which infects directly to Bursa of Fabricius, and once there is an infection, the whole organ look inflamed, red in color. There could be hemorrhages, and the organ is unable to produce a B lymphocyte. And if there is no production of B lymphocyte, one can understand that there could be immunosuppression, and the birds are susceptible for any further disease condition, maybe bacterial, maybe viral. So this organ is typically a very important lymphoid organ in case of poultry. This is well developed and mature in case of young ones, but once there is a aged condition, this Bursa of Fabricius will become rudimentary. So these are getting senescent or kind of atrophied in the later time point in the bird's life. So this is one such very interesting fact about the origin of B name that's coming from Bursa of Fabricius. Let's try to move on and see something different in terms of immune aspect. So when we discussed about the blood, we looked into the blood and defined the blood as a fluid connective tissue. So we can call the blood as a liquid connective tissue. So this could be a tissue because it's a collection of different kind of cells, and the blood as such is a very important point where people can use the parameter of the blood to tell about the disease condition or any abnormality in the human beings or also in animal. So there are terms which are related to blood which we can discuss, and few of the things which is very well documented in literature, and also one can emphasize that point that once there is an suffix emia, EMIA, that stands for blood. Anything which is related to blood is, and it is what it ends with emia. If you remember, we also discussed about inflammation. Any inflammation condition will eventually ends with TIS, TIS. Similarly here, anemia. Anemia stands for no blood. essentially, or anemia. Basically it stands for a lower concentration of blood cells or blood in the human body. So we can simply define anemia as a general term where the blood is limited or blood parameters are getting lower in case of individuals. So people are suffering from problems where the blood cells need to replenish back and all. So anemia is very common condition that happens in multiple conditions. There is a leukemia. So if you see emia is again there, so it is related to blood, and the front is leuc. Leuc stands for leukocyte. Leukocyte means blood cells which are white in color. So white blood is what you call leukocyte, white cells, white blood cells. So leukemia is actually the more number of white blood cell in blood circulation. So white blood cell concentration, which could be immature form of the white blood cell that comes into the circulation because of some uncontrolled proliferation, may lead to a condition where their concentration are quite high, and this is what in regular term is called blood cancer. So leukemia is actually more number of undifferentiated or immature white blood cell in the blood population. Then comes polycythemia. So if you see emia is again there, so it's blood. Poly means more, cythemia means cell. So more number of cells in the blood. We are not defining here whether it's a white blood cell or the red blood cell. It's simply more number of cells. Immature cells which are produced in the bone marrow cell, and these uncontrolled number of the cells in the blood is actually leading to a condition called polycythemia. So polycythemia is again term which is used to define more number of undifferentiated cell in the blood circulation. Then there is a term called hyperglycemia. Again emia is there. Emia stands for blood. Hyper means more. Glyce is standing for glucose. So more amount of glucose in the blood circulation is what you call hyperglycemia, which is actually diabetes. So hyperglycemia is a term which is needed by a clinician in order to decipher the level of glucose concentration in the blood. So hyperglycemia, again this is a hypoglycemia. So emia is again blood, hypo means less, and glucose concentration is what is defined by this term. So hyperglycemia is more and hypo means less glucose in the blood circulation. Then there is a term called septicemia. Again emia is there. Septicemia is a very specific condition where the blood is rich or fortified by toxins, and that can happen when there is a bacterial infection. Many bacterial species are known to produce toxins, and when there is a toxin concentration increase in the blood circulation, we simply call it septicemia. We also call the term called bacteremia, viremia. So this emia is again the term related to blood, and once somebody is saying bacteremia, that stands for bacteria in blood. Viremia means virus in blood. So these are the terms which are very critically used when we talk anything about the blood in general. Hyponatremia. So we understand that natrium is a term when we use sodium as a symbol. Na stands for natrium, which is actually a sodium ion. So if there is a lower concentration of sodium in the blood, the condition is called hyponatremia. We can also call it hypernatremia, which stands for lower or higher concentration of sodium in the blood circulation. So there could be multiple terms, but a student can always define it, that what are the things which are essentially defining the term. So one can easily break the word into prefix and suffix. The prefix can define some terminology, and the suffix can also ends with some kind of a meaning in terms of immunology. So these are few of the things which are related to blood. Let's move on and we'll see something more interesting. And one of the things which we have discussed earlier is serum. So serum is again a fluid part that we can isolate from blood, and there are procedure or the way the serum can be segregated out from the blood. So one can draw the blood, and this blood is without any anticoagulant. So the blood will simply clot once we leave the blood for a while. So once the blood is collected, we can just keep the blood for a while, the blood will clot. And once the clot blood is there in the tube, one can do a centrifugation. So this is a machine where we can put the tube and it will revolve to certain speed, and once there is a revolution of all those tube, so the blood and the cells and all will be segregated out, and what we'll get is actually the yellow color fluid on the top, which you call serum, and then there is an interface white buffy coat layer, which is actually all WBCs, platelets and other kind of cells, and then in the bottom we can get a red blood cell. So this is what we separate out. We can take out this serum, and this serum can be utilized for various purposes, and these are very, very important thing when it comes to immunology, because this serum actually contains immunoglobulins. This serum contains many proteins which are studied for immunology. They contain a lot of cytokines which we will study eventually throughout this course. So this serum is very important. The color of the serum is also defining some sort of health issues. When the serum is more yellow in color, that's an indication that some more amount of pigment is present, which is an indicative that the person might be suffering from liver disorder. That could be jaundice-like condition. If there is a redness in the serum, we can see and understand that there could be a lysis of RBC. So RBC might be destructed during the process also, it is possible. There could be a possibility that this serum become thick, very thick, that is possibility when a person is consuming large amount of fat. So this become a murky kind of liquid. It doesn't give a strong flavor of yellowness, but it may be a kind of a hazy color, which indicates that the serum is containing huge amount of fat, and that's an indication that the person might be having some cholesterol or some atherosclerosis or some kind of a dysfunction where the blood vessels or blood contains higher amount of fat or cholesterol or low density lipoprotein or any other thing. So it could be an indirect measure that the person might be suffering from some kind of condition. How we can segregate this serum with any other fluid? So serum actually came out after clotting, so we can understand, we can argue that there won't be any clotting factor there, and also fibrinogen, which is required for the fibrin formation. Just before the clotting, we form the fibrin, right? There are many factors which are involved in clotting. There are 13 factors. So these factors are absent, and also the fibrinogen, which is essential for the blood clotting, those proteins are absent, and therefore serum is devoid of any clotting factor. Mainly this contains very important protein called albumin and a globulin, which is actually the immunoglobulins, we called it. So Ig, different types of Ig which we have studied and which we will study in our discourse, that what are the different kinds of immunoglobulin, how they are important and all. This particular serum is used to diagnose a particular disease condition, because once this immunoglobulins are present in the serum sample, the presence of immunoglobulin against a particular disease can tell us about that disease outcome. Say for example, if a person is having an immunoglobulin which is reactive against HIV, one can say that the person might be suffering from HIV. If a person is having an immunoglobulin against influenza virus, we can say that the person might be suffering from influenza virus like other disease condition. So this is way people can identify a disease pattern or tell about the disease. We can also tell about whether the person is suffering immediately, recently started suffering from disease, or it's a chronic condition, say long lasting disease. So we can have acute and chronic condition where we can see that when we have an IgM concentration very high, we can call it the person is suffering from any acute infection. So IgM is an immediate raise in immune response in response to an pathogen entry. So IgM is acute immunoglobulin. say if there is a high upregulation of IgG, one can say that the person might be suffering from a chronic infection or long lasting infection. That means it's a very important tool to tell about the disease condition in case of human beings and also in other animal as well. People are using serum immunoglobulins to do research. Say if we want to identify a protein from a cell lysate, for example, if we want to try and look into NF-kappa beta or any kind of a protein which is important for DNA replication, we need to find out whether the protein is there or not. To identify that protein, we are using immunoglobulins or specific immunoglobulins against that protein. The process what we are using to identify that protein, we call it Western blot. So Western blot analysis, where we run the protein and probe with the immunoglobulin so that we can say that this is actually the protein which we are trying to look into or trying to decipher the biology for. So this is again a very important commodity when it comes to research. People are trying to look into protein, identifying protein based on immunoglobulin or any kind of immunoglobulins. There is an important tool in terms of therapeutic also. These are very important. So actually anti-venom production is also getting a serum. So what we are utilizing is actually the serum against particular kind of snake venom, and those are all utilized for treating the patient when somebody is getting a snake bite or any kind of thing. So anti-venom production, what we are getting as an anti-venom is actually is a serum. So the animal, any species which we have, which we can inoculate a small sublethal dose of the toxin, will sensitize with that toxin and will produce some kind of an antitoxin. That is what we are utilize for the therapeutic intervention. So antitoxin or antivenom, these are actually a part of the serum that's come from a different species of animal and all. So these are very important. We can use for diagnosis, we can use for therapeutics, we can use it to check if the person is suffering from any disease and all. So serum is very important when it comes to identifying a disease condition. We'll move on and we'll see that there is a term which, since we are discussing about blood, there is a term called erythrocyte sedimentation rate. So ESR in short is called ESR. This is not routinely used in the current scenario, but ESR was earlier used to identify a disease condition. So what's happening, so if we can collect the blood in with anticoagulant, so remember when we discussed about serum, we just collected the blood without any anticoagulant, so the blood will clot. But when we add the anticoagulant, blood won't coagulate, so blood will simply become a liquid. When you leave it for a while, this blood will start settle in the bottom because of the gravity, and later if you increase the time, you see that still the RBC become packed up in the bottom, and finally after some time we see a maximum settlement of the RBC that's happened in the bottom. So what ESR tells is actually the height of the clear plasma. So essentially we can define the term here called plasma. So plasma is the fluid that comes out from the blood sedimentation without any clot. So once we add the anticoagulant, what we'll get at the fluid form is what we call plasma. So essentially that length is what we call erythrocyte sedimentation rate, and that's measured after a particular time frame. The extent of ESR is what is utilized to check the status of health in human beings. So more ESR, more sedimentation of the ESR is happening when we have more of the protein in the system. If the person is having an inflammatory disease, any infection, that may lead to a condition where the erythrocyte or the RBC will settle very fast. So the rate of ESR become more high in disease condition. So this is one such way people have looked into the disease condition in the past. So ESR is again a very important parameter to check into the blood. In case of parasitic infection, since there is a histamine secretion in case of allergic infection or allergy kind of process, when it happens, when basophil is getting activated, the histamine production will increase in the blood circulation. And when there is more histamine in the blood, we can see that blood is settling more faster as compared to the normal condition. So these are all indication that the person might be activated some allergic reaction, and therefore basophil gets activated. So even if without checking the histamine concentration, one can check and one can presume that the RBC is settling very fast. That's meaning that the histamine concentration is pretty high in the blood circulation. So this is one such parameter which people look into when it comes to disease condition, called erythrocyte sedimentation rate. Since we discussed a lot about RBC, RBC is again a very important cell in the human body or in the animal which carry oxygen. So we all know that RBC, red blood cell, is a kind of a transporter for oxygen in all species of animal, but there are deviation. So once the RBC is matured, we call it enucleated. We are not calling it a non-nucleated, because they are non-nucleated stands for no nucleus, but enucleated stands for the RBC when they are formed from the bone marrow, they contains nucleus, but during the course of maturation they are losing out nucleus in order to create more and more space so that oxygen can be entrapped there. So the RBC is without any nucleus at the matured condition, just to accommodate more oxygen there. While in case of some species, for example in birds and amphibians, the RBC is nucleated. Particularly in camel, the RBC is oval shaped, because the camel blood, because they live in desert and therefore the blood is very thick because of maybe dehydration and all. So just to give a smooth flow of the blood become, or RBC become oval in shape. So these are all typical pattern which we will see in the animal world. Birds are actually, the argument could be the nucleated blood is RBC is there in the birds or other avian species because their bones are hollow and therefore they can trap the oxygen there itself. They have air sacs in their body which can trap more oxygen or more air into that, and therefore there is no additional requirement of needing some RBC which is without nucleus, and therefore there might be a possibility that these RBCs are all nucleated. The lifespan of RBC is actually 120 days, and after that period the RBC goes into spleen and get destructed and recycled into new form of the RBC. So RBC forms in the bone marrow and then it goes to circulation, there remain there for 120 days, and then after completing their lifespan they go to the spleen, which is called graveyard of RBC, and there it gets destructed and all their components such as hemoglobin, globin getting segregated out, the protein part, the iron part, and all those proteins which are there will recycle back and form, going through a salvage pathway to produce a new RBC cells. So this is a continuous cycle happening, and therefore the RBC maturation and their recycling is very important. The number of RBC is huge in human body. So there is a statement which is given in literature, is that a single RBC contains about 300 million hemoglobin molecules, and each molecule of hemoglobin can carry 4 oxygen molecules. So essentially one hemoglobin can bind to 4 oxygen molecules. Since there are 300 million hemoglobin, we can expect to have 1.2 billion oxygen molecule at any time in the blood circulation. So just consider the strength of this cell and how important it is in terms of carrying oxygen and all. It looks very beautiful in terms of its shape. It is biconcave structure. The color is red because of hemoglobin, and we can see that the blood coloration also varies in different species. We have mollusk, where you have snails and other species where the blood is containing copper instead of iron, and they are called hemocyanin instead of hemoglobin, and this color of the blood is blue because we know that blue color is coming from copper. We have blue vitriol, copper sulfate, H2O, which is actually a blue vitriol. Also, in case of arthropod, we have white coloration, so we called it hemolymph instead of hemoglobin because they are just white in color. They essentially contain zinc in excess as compared to what we contain iron in the blood. So essentially those all ions and the way individual animal behave can decide multiple things. So immunology is very interesting when it comes to the cell type which it carries. So let's see something more important. So since we are talking about blood, there is a way people make a blood smear. So how we can make a blood smear is we can take a slide, for example, it's a glass slide. We can put a drop of blood at the one of the corner, and then we can take a second slide, and then we can tilt the second slide maybe 30° to 45° angle, and then we can roll it little slightly backward and then move forward. So what will happen that we can have touched the smear the blood, and then we can pull it little forward and then pull backward. So in that way we can create a smear of the blood. That is what we call blood smear. We can keep it for air drying, and once it dries out, then we can just stain it and see what are the cells which are there. So to stain, what we can do is we can fix the blood smear with small methanol. So we can add methyl alcohol, and once we add alcohol, then we can put some kind of stain, and one of the stain is called Wright stain. We can add the stain and then wash with phosphate buffer slime with a normal pH 6.8, and then we essentially wash to remove all excess stain or kind of alcohol and all that's present there, and after drying out we can examine it inside the microscope, that what are the cells which are present, how much population of the cell, we can count the cell, and we can look into how this smear look like, and that can tell us a lot of things in terms of disease biology. So let's try to see that what are the things which we will see once you stain the cell population in the smear. So once you stain with Wright stain or Gimser stain, what we can see that there could be lot of different cells. We can see smaller cell which is called RBC, very small in size. The smallest cell is what we call platelet, and on top of platelet we see little larger cell population, that's an RBC, and the biggest cell which we'll see is all WBC actually in the blood smear. So let's start with platelet, the smallest, then RBC, and then we can see WBC. And in WBC also we can see the staining pattern. So some of the cells which we will see in the smear is stained with a neutral dye, and we can see that the nucleus is multilobulated. There could be 3 to 4 or 5 lobes there in the nucleus. That multilobulated cells is actually neutrophil, which is a phagocytic cell, very important cell, more abundant cell in the human blood. Then we have eosinophil, which is actually stained with an acidic acidic granules, which will stain and will give you a red coloration sort of thing. These are bilobed, so you can simply see 2 lobes of the nucleus. And then we have a basophil, where we can see a large dark blue or purple kind of a granule segregated throughout the cytoplasm. So these are basically cells which contains granule, and because of that region you can stain it out. Based on the stain you can say, and also based on the morphology of the nucleus one can segregate that these are eosinophil, basophil or neutrophil, looking into the pattern of the thing. Then we have without granular cells such as agranulocyte. So either we can get a lymphocyte or monocyte. Typically lymphocyte is actually the cells which we will discuss in this course a lot. T lymphocyte, B lymphocyte, we will cover this course all with lymphocyte. When we say lymphocyte, the nucleus is so big that we hardly see the cytoplasm. The whole cell look like a blue colored dot. Which is actually the nucleus. So nucleus is covering the entire cytoplasm. We won't see the cytoplasm per se. While in case of monocyte, we see the kidney shaped, horseshoe shaped nucleus, where we can see that there could be a very distinct pattern of the nucleus which is without any granule. So we can see that there could be a similarity between monocyte and eosinophil. It looks like a bilobed structure, but lobes patterns are different. But again, there could be a problem when we see in this smear but one can easily identify that once there is a granule, we can say it as an eosinophil. Without granule, we can say it's a monocyte. So these are all the cells present. Now, why we are discussing this white blood cell population here, that the presence of white blood cell population in the smear can tell you a lot of things. In general, earlier days when we were lacking the advanced technique or advanced say instrument, we used to just see the blood smear and say that the person might be suffering from a disease condition. For example, if we have more number of neutrophil in the blood, we can say that the person is suffering from a bacterial infection. So if doctor says and see that the report is showing some sort of more neutrophil, then doctor can understand that the person is suffering from bacterial disease. Let's try to prescribe some antibacterial or antibiotic. So this is what is very important when it comes to blood smear. We just simply look into the neutrophil concentration and we can tell that the person is suffering from some sort of condition. More lymphocyte in the blood circulation is an indicative of viral infection. So more lymphocyte when it comes, and if it is visible in the blood smear, there is no point of giving antibiotic because the person might be suffering from a viral infection, and if we give antibiotic there is no use because the viral infection it won't be treated with an antibiotic. Antibiotic is very specifically for bacterial infection. So lymphocyte increase is just tell you that the person might be suffering from virus, although this is not a very concrete parameter, but people do look into these blood smear picture very critically. These are not cytokine, we don't know anything detail about that, but simply looking into the blood cell, one can judge that what kind of things is happening. More amount of eosinophil is an indicative of parasitic infection. Say if the person is having some sort of parasitic infection, the eosinophil concentration will increase. We will see later once we see eosinophil concentration is very high, the typical cytokines which are produced by eosinophil cells actually give you an indication that the person might be suffering from parasitic or helminthic infection. So eosinophil is actually again an indicator of one type of infection, which is helminthic or other parasitic infection. Basophils are generally responsible for histamine production. So any allergic reaction, any kind of say allergen which is there in the air circulation, once person is coming in contact with an allergen, then we see that the basophil concentration is very high, which indirectly lead to more production of histamine, and therefore in allergic condition generally people are treated with antihistamines. Therefore these are also an indicative of some sort of thing. So we can generally look into the blood smear and we can judge that few cell type can tell us whether their high number or low number can tell us that the person might be suffering from a particular kind of infection. So the smears are important. Since we are talking about neutrophil, let's try to discuss about a very unique phenomena which we call netosis. Netosis is actually stands for neutrophil extracellular trap. It's a very unique phenomena, and I would emphasize that you just look into that very critically, because there are other cells which also behaves in a different way or the similar way. So what is happening that once there is an microbes infection in the human being or any animal, the microbes are coming, and since we are trying to see and understand that neutrophils getting upregulated in bacterial infection, so essentially neutrophils are getting sensitized and they can cause some kind of processes where they can engulf the bacteria and do multiple way. One such phenomena is where the neutrophil itself do certain unique thing, and what is unique is once they encounter a stimuli such as bacteria or any kind of stimuli, they can start decondensing the chromatin. So once they can start decondensing the chromatin, you see that there could be a fragmentation. The nuclear envelope become breakdown. The content of the nucleus such as histone protein and the DNA will come out into in the cytoplasm. So essentially all the cellular architect become disrupted. So all the DNA, histone protein and other things are all coming into the cytoplasm, and essentially after some time the cytoplasm, the cell membrane become burst out. So once the cell membrane become rupture, all the DNA content and everything is coming into the outside, and since the bacteria is there in the outside area, these DNA and the materials which are coming out from the neutrophils is getting trapped, is trapping all those pathogens which are there in the vicinity. So essentially these DNA and all can trap the extracellular pathogen or stimuli in order to nullify them. And this is one such phenomena where neutrophil can trap all those pathogens to nullify their effect, and therefore they can't produce a disease condition. So this is a host defense mechanism where a particular kind of a phagocytic cell, instead of phagocytosis, can rupture out and throw out its DNA to form a mesh or net like structure which can trap all the pathogen and try to nullify it. So these are very important phenomena that happens in the human body, and these are all immune cells. So immunology as such is a very interesting subject where we can discuss and try to see that what are the different phenomena that can happen. Instead of doing a phagocytosis, a particular cell which is known to do a phagocytosis can do a kind of a net like structure by just releasing out the DNA content into the extracellular area. So these are some of the interesting facts about immune cells. We will try to discuss something more in our coming classes. Thank you.